Think about the last time you saw a headline promising some massive medical breakthrough.
Oh, there's so many of those.
Right, and usually there's this underlying expectation of, like, vibrant, unstoppable life. I mean, when you hear a phrase like stem cell therapy, you probably picture a microscopic army of builders.
Yeah, that's the classic image.
You know, you inject these living, healthy cells into the body, they rush to the site of an injury, and they just start repairing tissue like a highly trained biological construction crew.
It is a very comforting image, and honestly, it's highly intuitive. In the medical field, this is broadly known as the living drug paradigm.
Okay.
The basic logic is pretty straightforward. You know, you take a living, healthy thing, you put it into a diseased environment, and, well, the living thing fixes what is broken.
Makes perfect sense.
Oh.
But then you look at the actual clinical landscape of mesenchymal stem cell therapy.
MSC therapy, yeah.
Yeah, right. MSC therapy over the last few decades. And suddenly, that beautiful, intuitive image just completely shatters. So today, we are taking a deep dive into a fascinating, honestly incredibly detailed perspective piece published in Cell Stem Cell by Donald G. Finney.
It's a fantastic paper.
It really is. And our mission today is to unpack a massive medical paradox for you all listening. What if, for some of the most severe inflammatory diseases out there, the secret to the cure isn't a vibrant living cell at all?
What if the mechanism of healing actually requires you to inject cells that are dead or actively dismantling themselves?
Yeah, which is just wild to think about. If you've ever found yourself frustrated by science headlines promising these incredible miracle cures that just, never seem to materialize in everyday hospitals, you are going to want to hear this.
Absolutely.
We're going into the messy, complex reality of biomedical engineering to understand why the whole one-size-fits-all approach to medicine is fundamentally broken, and more importantly, the radical new framework being proposed to fix it. So, let's start at the beginning. Why are we even talking about dead cells? I thought the entire point of a stem cell therapy was to keep the cells alive at all costs.
Well, that assumption is precisely what has created a massive bottleneck in the entire field. To understand the holdup, we really have to look at how regulatory agencies currently evaluate these therapies.
Like the FDA.
Yeah, like the FDA. The gold standard metric for an MSC therapy is viability. Specifically, regulations generally demand that more than 70% of the cells must be viable.
Meaning alive.
Right, alive,
Yeah.
after they are thought out for use.
So you look in the bag before it goes into the patient, and at least 70% of those cells need to be alive and kicking.
Yes, because the underlying assumption there is that these MSCs must act as, like, long persisting cellular factories.
Okay.
The idea is that they need to survive in the host's body, set up a permanent shop, and continuously secrete healing proteins and growth factors into the surrounding tissue.
It's like dropping a generic chemical factory into a war zone and just hoping it pumps out good materials indefinitely.
That's a good way to put it.
But wait, if high viability is the strict regulatory gold standard, and researchers have been adhering to this for decades, why are the clinical trial results so incredibly inconsistent? I mean, if the cells are alive, shouldn't they consistently heal the patient?
You would certainly think so, but the data paints a really highly confusing picture. The paper we're analyzing references this massive AI-powered analysis of 352 different MSC clinical trials.
Wow, 352.
Yeah, a huge data set. And what that analysis found was a completely chaotic landscape. Now, about 80% of the studies reported some kind of beneficial outcome.
Which sounds fantastic on the surface.
It does. But underneath that number, there was massive inconsistency. I mean, no single tissue source, no specific dosing strategy, and no testing method consistently outperformed any of the others.
So every single research team is getting slightly different results, and nobody quite knows why.
Exactly. And the reporting across these trials is incredibly fragmented. Nearly 70% of these studies lacked a proper potency assay.
Wait, meaning what? They didn't know how strong the dose was?
Essentially, yes.
Yeah.
They couldn't even accurately measure how powerful the biological drug was before injecting it. On top of that, over 40% didn't fully map out the surface markers of the cells they were using.
So we have researchers basically trying to force these highly complex, diverse biological products into a single, universal, living drug box.
Yeah, and that is what Finney calls the reductionist bottleneck. By assuming every stem cell has to act like a living factory, the field has kind of obscured a fundamental biological truth.
Which is?
In certain severe systemic inflammatory conditions like graft versus host disease or sepsis, the therapeutic benefit isn't actually coming from the living cells.
Okay, here we go.
The healing is being driven by cells that are intentionally dying.
Okay, we need to completely shift gears here because that is wild. Just to do a quick recap for everyone, we just established that the medical field has been totally obsessed with keeping stem cells alive. But if keeping the cells alive isn't always the answer, what profound signal is the body receiving when a therapeutic cell intentionally dies?
This brings us to what researchers call the apoptotic switch. But to really grasp this, we need to clearly define what apoptosis actually is.
Right, because it's not just regular death.
Exactly. It is not just random, messy cell death. It is programmed, highly controlled self-destruction.
So it's not a cell getting unexpectedly hit by a bus. It's a cell quietly, intentionally dismantling its own machinery from the inside.
That is a crucial distinction. When an MSC undergoes early apoptosis, it keeps its outer membrane intact so its contents don't spill out everywhere.
Oh, nice and clean.
Very clean. But it does something fascinating. It takes a specific lipid that normally lives on the inside of its membrane, and it flips it to the outside.
Like turning a jacket inside out.
Basically, yeah. This lipid is called phosphatidylserine, or PTD-SER for short.
PTD-SER. I'm picturing this PTD-SER molecule like a biological dinner bell or, a flag being raised on a sinking ship that explicitly says, hey, my mission is over, come eat me.
That is its exact function. PTD-SER is a universal eat-me signal in biology, and raising this flag triggers a process called efferocytosis.
Efferocytosis.
Right. Host immune cells, specifically the patient's own scavenging cells, like macrophages, recognize this flag. They use specific receptors on their surface to bind tightly to the dying stem cell and completely engulf it.
Let me make sure I'm wrapping my head around this. We are spending millions of dollars developing, culturing, and injecting these highly advanced stem cells. Just so the patient's own immune system can immediately treat them like an all-you-can-eat buffet.
Yes, but, and this is key, this is not just passive garbage disposal. Eating that dying cell is an active, radical reprogramming event.
Really?
Oh, yeah. When the host's macrophage consumes the apoptotic MSC, it completely rewires the macrophage's internal machinery and its function.
How does that work? Like, what is actually happening inside that macrophage once it swallows the dying stem cell?
To put it simply, the macrophage shifts its identity. So in a severe inflammatory disease, a lot of the patient's macrophages are stuck in what we call an M1-like state.
M1?
Right. M1 macrophages are aggressively pro-inflammatory. They secrete chemicals like IL-1 beta, TNF, and IL-8 that act like fire alarms.
So they're calling in more immune cells, causing tissue damage, creating swelling.
Exactly.
So M1 is essentially the madness state. It's actively fighting a war.
But when that M1 macrophage engulfs the dying stem cell, it triggers a distinct internal rewiring. It flips a master switch that shuts down the production of those fire alarms.
Wow.
The macrophage abandons the M1 state and transforms into an M2-like state. It starts pumping out anti-inflammatory molecules like IL-10, TGF-beta, and PGE2.
So it goes from M1 madness to M2 mending. It actively starts putting out the fire it was just contributing to.
It goes even further than just putting out the fire.
Yeah.
The physical act of consuming that dead cell forces the macrophage to synthesize specialized resolution molecules.
Resolution molecules.
Yeah. Specialized pro-resolving mediators, or SPMs, via an enzyme called 5-lipoxygenase. These molecules don't just dampen the inflammation. They actively command the surrounding tissue to heal, repair, and return to a normal state.
That's incredible.
They even create a feedback loop that makes all the other nearby macrophages better at finding and eating dead cells.
That completely flips the script. I mean, the dying stem cell isn't a factory at all. It's a sacrificial lamb.
A sacrificial lamb, yes.
The therapeutic mechanism isn't the cell doing the work itself. The cell is just the catalyst that licenses the host's own immune system to fix the problem.
The transition to cell death is a functional licensing event, absolutely. The biochemical act of dying orchestrates a masterclass in immune suppression.
Unbelievable.
The source material even points out that researchers have experimented with heat-inactivated cells.
Wait, meaning they literally cooked them?
Yeah, cells that are literally cooked until they are dead.
Yeah.
And they found that they can still quell inflammation in certain models.
Just because they were cooked?
Because the physical structure, that crucial eat-me signal, is still physically present to reprogram the immune system.
Okay. If the body is actively searching for that eat-me flag from a dying cell to trigger a healing response, doesn't that completely change how we should be storing and preparing these cells in the lab? I mean, we spend an absolute fortune trying to keep them perfectly healthy in freezers.
Oh, it completely reframes one of the biggest logistical debates in the entire field. The fresh versus frozen debate.
Right.
For MSC therapy to be a practical, off-the-shelf product that hospitals worldwide can use, you have to be able to freeze them. Cryopreservation is a logistical necessity.
Sure.
But traditionally, freezing has been viewed as deeply damaging.
Because it lowers the overall viability, and as we established, regulators demand high viability.
Exactly.
Yeah.
When you thaw frozen stem cells, they go through a temporary phase called cryolag, which lasts anywhere from 0 to 4 hours after thawing. During this lag, the cells are under immense stress. They suffer DNA double-strand breaks. They experience severe proteotoxic stress, sending out measurable molecular distress signals, like massive spikes in a protein called HSP70.
Basically, the cells are waking up from the deep freeze with a massive physiological hangover.
That's a great analogy. They are metabolically dormant. They also temporarily lose their ability to suppress immune cells because they have really low expression of an enzyme called IDO1.
Wait, IDO1?
Yeah, it's crucial for suppressing T cells. So their internal machinery is just focused on surviving the thaw. If you look at them immediately after thawing under the traditional living drug paradigm, they look like failing, useless cells.
But they can recover.
Yeah, usually a 24-hour post-thaw recovery period restores their cytoskeletal and metabolic activity perfectly.
I'm not starting to see where this is going. If the goal for something like systemic inflammation is to trigger the immune system by feeding the body-dying cells, couldn't that massive physiological hangover from being frozen actually be a therapeutic advantage?
Well, let's connect that to what happens when you actually inject these cells. There is a phenomenon known as IBMR.
IBMI?
Yes, the instant blood-mediated inflammatory reaction. When you infuse stem cells directly into a patient's bloodstream, they encounter the host's active immune cascade.
Right.
This triggers a massive, rapid reaction. The blood starts clotting around the cells, complement proteins attack them, and inflammatory signals spike. It's essentially a bloodbath that can destroy a huge fraction of the infused cells before they ever leave the bloodstream.
A bloodbath. Traditionally, scientists must look at that and think it's a catastrophic failure, right? The bloodstream is murdering the precious living drug before it can even do its job.
Under the old paradigm, yes, it's considered a complete disaster.
But wait, based on everything we just uncovered about the immune system needing a sacrificial lamb, could this bloodbath actually be the intentional catalyst? Like, could the trauma of being frozen, followed immediately by the violent shock of the bloodstream, be the exact perfect recipe to force those cells to raise their eat-me flags and trigger that healing macrophage response?
That realization is exactly the leading edge of this new framework. In contexts like systemic inflammation, the bloodbath isn't the failure of the drug. It is the necessary in vivo crucible.
Wow.
The stress of the freeze primes them, and the shock of the blood pushes them over the edge into controlled apoptosis. The death of the cell is the therapy.
That is genuinely mind-bending. It makes you wonder how many failed trials were actually working exactly as nature intended, but the researchers just didn't understand the mechanism.
Absolutely.
Let's do another quick recap for everyone listening just to consolidate this. We thought stem cells had to be alive to work.
Right.
But we found out that in diseases with crazy high inflammation, the cells dying, specifically raising that PD-Sercer flag, is what actually fixes the problem.
Yep.
So let me play devil's advocate for a second. If dying cells are this incredible biological switch, should we just be intentionally killing all stem cells before injecting them for every single disease?
We have to pump the brakes on that idea immediately.
Okay, why?
This is where the precision aspect comes in, because context is absolutely everything. The dead cell hypothesis is not a universal truth.
Got it.
If you drop a dead cell into the wrong disease context, the results aren't just ineffective, they are disastrous.
So what happens if you drop a dying cell into an environment that actually desperately needs a living builder?
The paper refers to this as context failure. There are diseases where you absolutely require viable, highly metabolic, persistent stem cells.
Like what?
A great example is conditions requiring tissue regeneration or restoring a physical barrier in the body, like acute respiratory distress syndrome or ARDs.
Which was the focus of the START clinical trial mentioned in the paper, right?
Yes, exactly. In the START trial for ARs, or in models for things like osteoarthritis or ischemic injury, the therapeutic efficacy is intrinsically linked to the cells surviving.
Okay, so they need to stay alive.
They need to be robust, They need strong mitochondrial resilience. They have to express high levels of factors like HIF-1 alpha, which helps them survive in low oxygen.
Right.
They need to actively secrete factors that build new blood vessels and repair physical tissue. In some autoimmune diseases, the stem cells even have to wear a molecular disguise just to hide from the host's natural killer cells so they can persist long enough to heal the damage.
I like to think of this like a construction site.
Right.
If a bridge is structurally compromised, let's say that's osteoarthritis, you need living workers to show up, pour concrete, and rebuild the pillars.
Good analogy.
But if the bridge is currently engulfed in flames like in severe systemic inflammation, sending in more workers just adds bodies to the fire. You don't need builders, you need to trigger the sprinkler system.
That's a fantastic way to visualize it. If the disease needs structural rebuilding, you need living cells.
But what actually happens biologically if you accidentally use dead or dying cells in a disease that needs structural repair?
If you put dying cells into a host environment that lacks the capacity to clear them efficiently, meaning the local macrophages are either too overwhelmed or too sparse to eat the dying cells quickly, the stem cells progress from that clean, controlled early apoptosis into what is called secondary necrosis.
And necrosis is the messy, uncontrolled kind of cell death.
It is biological chaos. The dying cells eventually lose their membrane integrity. They literally burst open, spilling all their internal cellular contents into the surrounding tissue.
Yikes.
Yeah, and this releases what we call damage-associated molecular patterns, things like ATP, HMGB1, and SO100 proteins.
That sounds like molecular shrapnel.
That's exactly what it is. Instead of presenting a polite eat-me flag, these ruptured cells throw biological shrapnel everywhere, which actively trips the body's panic alarms.
Oh.
Instead of triggering the mending macrophages, it triggers a pro-inflammatory danger pathway. This recruits aggressive immune cells, increases tissue-damaging chemicals, and causes the release of toxic extracellular traps.
So instead of putting out the fire, you just dropped a hazardous waste spill right in the middle of it.
And this context failure actively exacerbates systemic inflammation. It has been shown to make conditions like asthma, acute lung injury, ulcerative colitis, and lupus significantly worse.
Wow, that is a huge risk.
Interestingly, even actively blocking apoptosis in cells that are supposed to die can ruin a therapy.
Wait, how so?
The paper details a study on allergic airway inflammation where researchers used genetically modified stem cells that were literally incapable of undergoing apoptosis.
They made them immortal, essentially. And what happened when they injected them?
They completely lost their therapeutic efficacy. The disease didn't get better. But when the researchers pre-induced apoptosis, forcing them to die before injection, the therapeutic benefits magically returned.
Wow. So the therapy isn't a static object. It is a delicate, intricate dance between the specific state of the product, whether it's actively building or actively dying, and the specific microenvironment of the patient's disease.
Precisely.
Which leads to the ultimate hurdle here. Since forcing everything into a living drug box is clearly failing, how does the medical field actually move forward? I mean, how do you manufacture therapies when the finish line changes depending on the disease?
This is where Donald Finney proposes a radical shift to what he calls the precision product ecosystem.
Okay.
We have to stop treating the mesenchymal stem cell as a single universal entity. We need to start treating each cellular preparation as a unique biological drug, custom-tailored to a specific mechanism of action.
What does that ecosystem actually look like in a practical, real-world laboratory?
It has to start with the donors themselves. There is huge intrinsic diversity among donors. Not all stem cells are created equal.
Right.
We need to actively screen donors for specific genetic profiles to predict exactly how their cells will behave under stress.
Walk me through a scenario. Let's say a patient comes in with GVHD or sepsis.
In that scenario, we know the patient needs rapid immune resolution. We need the sacrificial lambs.
Right.
So we would screen our donor registry for individuals whose cells naturally express high levels of specific stress response markers, like a gene called TSG6.
TSG6?
Yes. High TSG6 means their cells are naturally more prone to undergoing rapid apoptosis. We would purposely use cryopreservation protocols intentionally designed to induce that death signal. When they are thawed and infused, they are perfectly primed for the host's immune system to consume them.
Tailor-made to die efficiently.
Exactly. But if our target is a myocardial infarction, a heart attack, or peripheral artery disease.
Right. In those cases, the target tissue is lacking oxygen. It is dying. We need cells that will survive at all costs and build new blood vessels.
We need the architects.
Right.
So we go back to the registry. We screen for donors whose cells have a specific resilience profile. Maybe they express high levels of a marker called TWIST1.
Twisty one.
Yeah, TWIST1 high cells are highly resistant to stress and naturally pump out massive amounts of growth factors. We would manufacture them using fresh culture techniques, or mandate a strict recovery period after thawing to guarantee maximum viability and ensure they act as vibrant cellular factories.
The source mentions a framework to help map all this out, the CLAP scale, right?
Yes, the Clinical Indications Prediction Scale. It's a conceptual framework that maps stem cells along a functional continuum rather than treating them as a binary pass or fail.
Okay, how does it work?
On one end of the spectrum, you have highly viable, resilient cells actively building blood vessels, the pro-angiogenic side. On the completely opposite end, you have cells transitioning into a pro-resolving apoptotic state to fight severe inflammation.
So it's a sliding scale.
Exactly. Efficacy isn't just asking, are they alive? It's asking, where do these specific cells fall on the continuum? And does that perfectly match the disease we are trying to treat?
That is a beautiful biological theory, but we have to talk about the real-world regulatory nightmare this creates.
Oh, it's a massive challenge.
Right. I mean, if regulators like the FDA are currently demanding greater than 70% viability across the board, how on earth do researchers convince them to approve a batch of cells that have been intentionally stressed out and primed to die? This completely shatters the established regulatory playbook.
You're hitting on the core problem. It requires a massive structural shift in how we report and regulate biology. We have to build a centralized registry that tracks absolutely everything.
Everything.
Everything. We can't just rely on viability metrics anymore. We must mandate deeply detailed documentation on the specific tissue source, the exact culture media used, the expansion platforms, and deploy highly standardized potency assays that measure the specific function we want.
We'd even need to track that bloodstream bloodbath we talked about earlier.
Yes, absolutely. Future standardized reporting will have to include measures of IBMR-related parameters. We have to track the immediate immune interactions in the bloodstream like platelet consumption or clotting factors and mathematically correlate those events with clinical outcomes. We have to stop viewing product heterogeneity as a nuisance or background noise and start utilizing it as a deliberate design parameter.
Okay, we have covered some incredible ground today. Let's pull all of this together for everyone listening. For decades, stem cell therapies have stalled because we've been forcing a messy, beautiful, highly complex biological reality into a neat, marketable, living drug box.
Yes.
We blindly assumed that being alive equaled being potent.
But by embracing the reality that cell death and cellular building are just different, equally valuable phases of a functional continuum, we can unlock an entirely new level of precision medicine.
We discovered that in cases of systemic inflammation, injecting dead or dying cells triggers the body's eat me signal. This flips the patient's immune cells from causing madness to mending tissue.
Right.
But we also learned that if you drop dead cells into an environment that desperately needs structural repair, you trigger a toxic danger pathway that makes everything worse. High viability isn't always good, and cell death isn't always bad. It is entirely 100% dependent on the microenvironment of the specific disease.
By abandoning the simplistic one-size-fits-all manufacturing model, we can finally build a true precision ecosystem where the intended therapeutic mechanism dictates the entire manufacturing pipeline, from donor selection to the freezing protocol.
Which leaves us with one final, lingering thought for you to mull over. If our bodies have evolved to utilize the death of a therapeutic cell as a profound, orchestrated signal for healing and immune resolution, what other medical interventions are we fundamentally misunderstanding right now? Are we looking at other advanced treatments through an overly simplistic living versus dead lens? Maybe the future of advanced medicine isn't just about figuring out how to keep things alive, but learning how to finally master the art of a perfectly programmed death.